Dear students, this video is talking about a mutation. Mutation is a permanent change in the DNA sequence of an organism's genome. Mutations can occur naturally during DNA replication or be induced by environmental factors like radiation, chemicals, or viruses. Let's start. Gene mutations are changes in the DNA sequence that can affect how gene functions. Point mutations involve changes in a single nucleotide and are of two main types. The first one is the substitution mutations. One nucleotide is replaced with another. And the other type is frame shift mutations. This often results in a complete and usually non-functional protein. The first type of substitution mutation is the missense mutation. This point mutation, where a single nucleotide change in the DNA sequence results in the substitution of one amino acid for another in the protein. Let's look at this example. As we can see here, the cytosine, this nucleotide, substituted by adenine here. So the whole amino acid changed. It was proline, now it is threonine. And this impact ranging between harmless or harmful. Now, may this impact change the structure and the function of protein. One example of this, the sickle cell anemia, when the hemoglobin protein change one of its amino acid. And now the second substitution mutation, which is the nonsense mutation, which is a type of point mutation where a single nucleotide change in the DNA sequence creates a premature stop codon. What does that mean? No more translation, incomplete protein, shortened protein that mostly is non-functional, and it will affect the biological process. An example of this is the Duchenne muscular dystrophy, DMD, or certain types of cancer. Moving to the third type of substitution mutation, which is silent mutation. That is a type of point mutation where a change in a single nucleotide does not alter the amino acid sequence of the protein. Let's see here. As we can see, instead of adenine in this original code, we have here thymine. And both of them, they encode to the same amino acid, which is valine. That's why we call it silent, because there is no effect on a protein function. We call this type of code a redundant genetic code. As a summary for substitution mutations, we have the nonsense, which will create a premature stop codon, and that means we have a shortened protein, non-functional. We have the missense, by changing one amino acid, also will lead to change in the protein structure, which will affect the function also. And the silent mutation, which will encode to the same amino acids. That means there is no change on the structure or the function of the protein. And now to the second type of point mutation, which is frame shift mutations. Frame shift mutations is a type of genetic mutation that occurs when there is an insertion or deletion, adding or removing of one or more nucleotide in a DNA sequence. Here we have insertion. Listen, since the genetic code is read in triplets, codons, which each specify an amino acid, this shift alters the entire downstream reading frame of the sequence. As a result, it changes every subsequent codon, which can lead to the production of a completely different and usually non-functional protein, as we can see. So the only codon, the first codon, will give the same amino acid, but the translation happening now, all the sequence will change, so we will receive different proteins. The other type of frame shift mutation, which is frame shift deletion. Again, one or more nucleotides are removed from a DNA sequence, and that causes a shift in the reading frame, altering the grouping of a nucleotide into codons. As a result, the downstream amino acid sequence is disrupted. So, again and again, we will receive a non functional protein. To summarize it, the frame shifting mutations, its impact is greater than the substitution. And you can use the amino acid wheel in order to practice the deletion and the insertion. As you can see, the only amino acid stays the same is the only one, and the whole sequence changed for both deletion and insertion of nucleotides. Also, this table will help you to summarize what you learned about the pointer mutation, starting by missense, nonsense, and you can add also the silent mutation with some examples. Also for a frame shift, the deletion, insertion, and another type of insertion by duplication, by inserting another 3 nucleotides, or expanding mutation through several generations with, as I mentioned, some examples. Thank you.